introduce Vincent Debat from the French National Museum of Natural History, please.
Good afternoon to everybody. Can you hear me well? Okay. So it's a great pleasure to be here today and to have this opportunity to present our project on the evolution of Morpho butterflies, gorgeous animals. So I'm Vincent Debat. I am professor at the Museum of Natural History in Paris, and I'm also the curator of butterflies. So these are my favorite animals. Morpho butterflies are likely the most famous butterflies ever. They're really famous for their blue iridescent colorations. So they've been heavily, and they are still heavily collected and sold. And yet we don't know that much about their ecology and their evolution. So that's one of the main aims of our project. So what do we mean by iridescence? So these are two images, or this one from the field in Peru, and this one from samples in the collections. So it's really strong variation in color and brightness, depending on the angle of illumination, but also the angle of observation. Really striking phenotypes. So as you can see on this image, so I made this using one single image per species. So there are 30 different species of Morpho. Morpho only live they are a neotropical butterfly, meaning that they only fly in the forest of the Central America and Southern America. And so as you can see, there's a huge diversity of phenotypes, and not all Morpho are blue. So one of the questions we are tackling in this project is why do we observe this diversity of phenotype? So this blue color in Morpho, if we zoom on a particular species, we see that this is a structural coloration. I mean, this means that there are no blue pigment in Morpho. And you cannot dye clothes using Morpho powder, for example. And if we zoom on the wing, so it looks like the roof and with tiles on the roof. And these tiles are actually scales covering the wings of. Actually, all butterflies are shaped the same way. And we can zoom again and again using SEM. And we can see at the highest magnification these exquisite nanostructures that are shaped like a Christmas tree. And these tiny structures, they act as nano mirrors, and they will reflect light and produce this really incredible light effect at the macro scale. So we are interested in the diversity of these nanostructures. But as I said, we are interested in understanding the diversity of coloration. And so this begs the question of what are the evolutionary drivers of the evolution of this iridescent in Morpho? Why aren't all species blue? And why, even between the blue species, we can observe this diversity? So Morpho also interesting in terms of ecology. Some species fly very high in the canopy, so in an open environment with full of light, while others fly very low in the understory, and the understory in the Amazonia is really cluttered, really dark in light. Some species fly super early in the morning. So one particular species flies only between 6 and 6:25 in the morning. So it's really low in terms of light conditions, and others fly at different moments of the day. So lots of variation in terms of ecology and light environments. So we've got one hypothesis about the evolutionary drivers of this iridescence, and this is what you can see here. So what I've done here is just turning the same animals upside down. You're seeing the ventral side, while the blue side is the dorsal part. And this creates really strong visual effects when these guys are flying. So when the dorsal side is exposed, you can see the bright blue things, and when the ventral part is exposed, then they just basically disappear. So you see really bright flashes, and with a period where you cannot see anything anymore. So combined with a fast erratic flight, we think that this blue coloration makes these guys really difficult to capture. And this, I can testify from my experiments of human predators. They're really difficult to catch while easy to spot from far. And we also have data suggesting that birds have the same difficulties to capture them in flight. So our hypothesis here is that the evolution of blue iridescence is related to natural selection imposed by predators. And under this hypothesis, we have this prediction that we should have a tight coevolution between blue iridescence and flight performance. Okay, first hypothesis. Second hypothesis is sexual selection. So this is one species, Morpho retenor, and this is another one. So in the first species, a super strong sexual dimorphism with the male being super blue and the female is not blue at all. But in other species, really low sexual dimorphism. But we think that sexual selection might be involved, and in all species, males will fight each other, and they actually will be attacking any blue object they will encounter, which makes our life easier because we can attract them with blue lures, and they can, they're easier to catch this way. Okay, and under this hypothesis of a major role for sexual selection and species recognition, we predict a tight coevolution between iridescence and color vision in these butterflies. And this is exactly the structure of HFSP project. So we have one part investigating the flight and flight aerodynamics, one part centered on quantifying iridescence, and one part centered on quantification of color vision. And all that is integrated by our evolutionary ecology approach. So this starts by bringing the physicist in the field and to meet the butterflies in real life and acting in the natural habitat. And it was good fun at the same time. Okay, so first part of the project, quantifying iridescence. So this is the job of our Colombian colleagues. And so they've been measuring and investigating the wings of these butterflies on all scales, so investigating the disposition of scales, quantifying far-field reflection, investigating, measuring the nanostructures at the surface of the scales, and also quantifying polarization. So as I said, iridescence is a variation in reflectance depending on two angles, illumination and observation. So this makes iridescence super difficult to measure because you need to combine variation of these two angles, so leading to the quantification of more than 600 spectra per species. Really time-consuming and super difficult task. So I'm just presenting one result on this iridescent part. So our colleagues measured the structures on the wings on various angles, on various aspects, and they applied multivariate statistics to investigate the structuration of this variation across the genus, and they identify 6 different clusters of morphologies that seem to be associated with different reflectance properties. So integrating these morphological measurement with modeling of these nanostructures leads us to get a pretty nice understanding on the link between morphological structures on the wing and the optical properties. And yeah, I invite you to check the posters, one of the butterfly posters that you will see. Okay, second part of the project, the analysis of flight. So as I said, we suspect that there's a coevolution between escape flight and iridescence. And for that, we need to quantify flight, which is, again, super difficult. So we measured various things. First thing, we are interested in the escape flight and how these guys escape the predators. So we try to do that, to investigate that in the field. We also investigate kinematics and wing deformation. Just to give you an example, so this is a movie with a grad student, Raphaël Dupillier. You can see he's moving the blue stuff, and the butterfly is coming. I don't know if you can see it. So he's mimicking an attack by dropping the net at the butterfly. And we are filming the scene using various cameras, so from different angles. And we track the butterflies on each of these movies, allowing to reconstruct the trajectories in 3D and to investigate the different flight parameters. And in particular, comparing the pre-attack and post-attack, which is the escape part. So I'm just showing the details on one particular species. So this makes this spaghetti-like set of trajectory. We can make sense of it. And in particular, we measured erraticity before and after the attack, also speed. And the interesting stuff we found is that there's a tight negative correlation between the two, suggesting that the attack butterfly increased the erraticity of the trajectory, but this comes at the cost of a reduced speed. So this suggests that erraticity is particularly important in the escape flight of these blue butterflies. So what is the biggest picture at the scale of the whole Morpho genus? So we are beginning to get the data ready. I'm just showing some new, very brand new results that my colleagues haven't seen yet. But we found that there's a really tight association between iridescence and erraticity of the trajectories across species, with the brightest and most iridescent species showing the most erratic flight. So this is perfectly in tune with our expectation, which points at the possible effect of predation driving the evolution of and the coevolution of flight and iridescence. Now, in terms of color vision, so this is big team with some American colleagues, some colleagues in France, and some colleagues in Slovenia. So two main approaches for this analysis of color vision: a molecular analysis of photoreceptors, which are called the opsins. And the first part of this analysis of opsins relies on the genomic analysis of sequences and the analysis of selection. The second approach using immunohistochemistry to identify the expression, the localization of expression of these photopigments throughout the eye of these butterflies. The second main type of approach is developed by our Slovenian colleagues, so mostly based on electrophysiological analysis of color vision, so intracellular recording of the activity in the eyes of these butterflies to test the effect of different wavelengths on the sensitivity of the eyes of the different species. So I invite you to check the poster of Gregor Belugic, which is packed with really nice new data. So to focus on only one main result, on the molecular part of the story, so we sequence the different opsins, and we've shown that there are two duplications of the long wave opsins in these butterflies. And analyzing the variation in amino acid composition, we detected a few changes that are associated with shifts in blue sensitivity in these different species. We also quantified or investigated the signs of selection along the sequences of these proteins and detected some effects associated with differences between blue and non-blue species, but also between canopy and understory species. And so to summarize that, it suggests that there are strong adaptive there's a strong adaptive evolution affecting the photoreceptors of these species. I will end up with an interesting coevolution between wing iridescence and vision, in particular using these two sister species. So they are really closely related species. Morpho Eugenia, that's the one that flies only between 6 and 6:25, so really in the dark, while its sister species flies later on during the day, so in full light conditions. So you can see there are different colorations. So Eugenia is paler than its sister species. But when investigating the color vision using electrophysiological data, they seem to be fairly similar. So the question is, are they really of the same color? And if we combine their reflectance, their color, with the irradiance spectra obtained, the light that they encounter at the different moment of the day where they are flying, what we see is that the resulting color is strikingly similar, meaning that they have the same color at these different moments of the day where they fly. And this suggests that we might be facing a nice case of evolution of color patterns on the wing that might have balanced the shift in light conditions during the day, so it might be a case of sensory drive evolution. And I invite you to check the third poster on Morpho that will give you some details on that. Okay, to conclude, these Morpho butterflies are super interesting for various reasons. Really an interesting thing is that they nicely captures different general elements in phenotypic evolution. First one is that they provide nice cases where same selective pressures will jointly affect various phenotypic traits. And this allows us to study these cases of adaptive syndromes. Second thing is that these allow us to investigate the relationship between the evolution of one trait and the consequences on the evolution of other traits. So these are the cascading effect among traits. And with that, I thank you for your attention. I invite you to check the posters.